Yes, thank you, Bill. We can definitely be a poster child for biolabs, for Danforth, for Yale Ventures, where Yale spin out companies. So all have been fantastic and instrumental in the progress that we've had to date. We are a pre-seed company, and I was formerly a Yale faculty member up until about 8 weeks ago. Took the full leap in without much of a safety net, but because I believe deeply in what it is that we're trying to accomplish at Revalia. Perfect. And what it is that we're trying to accomplish is really a new play on trying to transform the process of how we do biomedical innovation. We've heard about so many amazing technologies here today and yesterday too, but there's so many challenges in the process as it exists now. And in particular, we like to summarize it by saying this system as it exists doesn't fail very well, right? So we all know this pipeline. The problem is it takes an incredibly long time to fail in the setting that we most care about, which is a human with a disease of interest, if it's a drug, for example. And the consequences of that failure are massive, right? Obviously, we put patients' lives at risk, but there's massive economic consequences. It can reach $1 billion, right, depending on the stage of the development. And the failure isn't useful in the beginning stages when we could use failure, because we often start with the wrong systems. We don't begin with the patient, with the end in mind, with a highly variable patient population. We begin with what's available to us. So at Revalia, and what we've done in my lab for the last 10 years is come up with a new thesis, which that if we can recenter on a new system, which is donated human organs that are not usable for transplantation, but can be used in research settings, then we can transform this process. Because first and foremost, we begin with the end in mind. We start with a highly variable human population. We can go incredibly fast, so we can actually sort of start with the clinical trial in the beginning, so we can be very agile about it. And the whole thing is we're not putting patients' lives at risk, so we can actually, it's allowable to fail, and we can transform the catastrophic failure in a clinical setting into a catalytic failure in this setting. And while doing so, we can actually meet the needs of donors' families who also suffer a catastrophic loss when they're told, I'm sorry, your loved one's organs weren't good enough for transplant. And the reality is that for a patient that passes away in ideal conditions for donation, over 90% of the time, that's what those families are told, because that person has cancer or other things that are rule outs for transplant. But they often desperately want to have something meaningful happen afterwards. If your loved one loses from cancer, you want to continue the fight and help in some way possible. So what we're enabling is the ability to receive those organs at any time of day or night, just like we would be able to do in a transplant setting, bring them back to life, and prove that an organ doesn't have to be transplantable to be transformative. So what does that look like? Well, it starts with these systems. So what you're looking at is a machine perfusion that's actually used clinically in the transplant setting. The way I want you to think about it is a mechanical patient, right? So this mechanical patient, it actually has heart, lungs, we've modified to have kidney, we control nutrition, and it has a brain that acts as an algorithm so that when we bring this organ back to life, unfortunately the video's not playing, I'll show it to you all later if you want to see it, but what you would see there is the liver being reperfused, coming back to life, and when it does, it'll do things that livers do. So it'll synthesize proteins, it'll detoxify, it'll also propagate the underlying injury associated with the disease, which in this case is sterile inflammation associated with fatty liver disease. We've developed with some partners the ability to go out to 5 days so that you can study that dynamically and evaluate not just disease pathophysiology, but the ability to test interventions. And we do that by leveraging our real expertise, which is the ability to sort of speak the language of the organ and extract all of the knowledge using all of the cool tools that we have, from 3D rendering of organs to quantitative histology, multi-omic technologies, and even could be a breeding ground for all the amazing new spatial omics and technologies we've heard about here today. The really cool thing is that you can now functionally analyze the organ as well. So this is an example of some work we did in a science paper several years ago, testing the ability to improve bowel quality in an individual organ. So what we do is really a partnership play. So we are a catalyst company that is seeking to really accelerate and de-risk high-quality innovations with partners. What our expertise is around is being able to design these high-complexity experiments, execute them, and deliver high-quality data. And we've got a full-stack technology team that is behind this, building all of this out. This is an MVP version that started at Yale, but we now have a full tech team building out, or actually have released our second version. And with that, what we're able to do is really fill a gap between animal and human clinical trials, and leverage the inherent complexity and variability of human systems, which is the point. We need that. That's what our patients look like, so we need that to be able to develop the appropriate therapies. we can be forced to think about some of the late-stage problems like delivery and other scale-up bottlenecks that, if we wait until the end, we can't actually scale that up, or we can't actually deliver it effectively into a patient. So that's why this becomes a catalyst. And I've sort of talked about in the context of therapeutics, and that's where we started for, but it really isn't limited to that. It really can apply to different, diagnostic tools or med tech devices, and we're really exploring across the landscape. The challenge is being able to scale it up and deliver really on the speed. And so that is what led us to leave Yale, because it wasn't possible to really solve those problems in the academic setting. And so we've built now a full stack development team. It's a flexible staffing model. So this is, I'll finish fast. This is our 6th team. We have 10 teams behind it building new software and hardware. We have an amazing perfusion team that's breaking new ground, including recently doing the world's first triple liver perfusion for research simultaneously. And we've built out a bunch of relationships to build a network out to do this. We now can work in lung, kidney, and liver, but have ability to expand into heart and pancreas, expanding into cancer, multiple different style of perfusions that we can do, and multiple different types of injury and disease models. We have an incredible team that we've assembled, not just our executive team, but advisors who are really guiding the way on making not just very exciting science and technology, but really a very scalable business that can be around to make sure that we can drive as many innovations into patients as we possibly can. Thank you very much.